The topic which we'll be covering today is systematic outbreak investigation in sheep and goats for effective prevention and control. So this is part of for our activity in terms of the Azadi Ka Amrit Mahotsav India, the 75 celebration we are having. And this is the week 58th. And the theme of this is the animal healthcare for better production. For today's discussion, I would like to cover a very basic of understanding epidemiology of a disease, not only for the sheep and goat, for generally for all the species also, and what are the steps to be followed. And the topic which we'll be covering is which can be done at the field level. Make it a point that the things what we cover is completely can be done by the field veterinarian at the field level with limited facilities or resources at his disposal or at his or her disposal in the field. Any epidemiological studies, especially one thing is very important to know. In India, we have to accept the fact that we are not, very good expertise in the epidemiology subject as such, starting from UG and also in the practice in the field. And as we were discussing with our director also on how important it is in terms of the vaccination and treatment is one aspect, but the other most important aspect, which we can prevent and control, is understanding the epidemiology of a disease. I always start with this particular slide of breaking down the definition of epidemiology into different components. When you call it a definition, is study of distribution and and determinants of health related states in specified population. So the most important aspect when you talk of epidemiology, without population, the study is incomplete. And most of the cases we see that we talk about individual cases. We don't talk of the population. So that is one important component to be kept in mind when you are understanding any epidemiology of any disease. Most of the times, the reason for not understanding the epidemiology is we try to not follow a proper approach to study the disease in terms of the outbreaks or also retrospective studies or any other study design we adopt. When you talk of study, it's always very simple W's of epidemiology, what they say, when, where, and who point is very important. And in analytical, we also try to address how and why. And we try to train people and also ask people at the field level to see that how best they can do the descriptive part. At least that can be done with a lot of whatever facilities they have. The analytical component can also be taken up. It's not that difficult, but it can be supported by the institutes like ICR. And when you talk of distribution, it's very simple thing. When you talk of the disease, we have to talk of the space and time component. Like when you talk of the determinants, we have this all your triad of host, agent, and environment. So the health aspect, again, we talk about the infectious disease. The other most important aspect of these health point of view of animals is non-infectious diseases, and that is completely lacking. At least we do the health, the infectious disease aspect. We try to do a lot of studies in that, but non-infectious disease, again, one important component to be done. And in terms of the sheep and goat, again, is very important, like what kind of feed they are access to and why. Maybe I'm not covering in that as an example, but that is very important factor to consider when investigating outbreaks. What are the non-infectious causes? That's the first thing we have to do. I'll cover in the next slide. As I said, the host. And if you take this definition without doing this study in a population, it is incomplete. So that's one important point you have to remember throughout this lecture and also after this lecture, after this webinar, to do systematic study or the outbreak investigations by considering the population. Here, the population can be target, can be your village where the outbreak is happening. So the very purpose of understanding any epidemiology in terms of outbreak investigation or any other studies, so identify the cause of the disease, and the most important is how best we can prevent and control it. If you identify the cause, if you do diagnostics, if you identify the source of infection, but if you don't adopt a systematic control strategy to control the outbreak or prevent it for the future occurrence, unless we do that, it's incomplete. So that's one important thing we have to remember always. And what is the role of epidemiologists? And when I say epidemiologists, like all the veterinarians in the field are epidemiologists in that sense, because unless we have a specific designated post, we need to think that we can do minimum field level epidemiology thing. So we always, maybe most of you may agree with my point of, when we go for outbreak investigations, we try to study the disease, animals with disease. And we forget most of the times the animals without disease. And one important component when we try to understand and do investigations in field, we have to also collect the data from healthy animals. Then only we can compare the difference between diseased and non-diseased animals. So there lies the very, very critical component. Like when we do this systematic outbreak investigations and try to see the exposure in animals, with the disease, and the exposure in the animals without the disease, so that we can say risk factor point of view. So this thing is very important when you are trying to study the disease. And I repeat this point of studying the disease with animals with the disease and without disease. And I'll cover those points in detail when I go for the steps in outbreak investigation. And when you start any outbreak investigation, one thing, as I said in the previous slide also, you always need to start with whether the problem you're studying is infectious or non-infectious. Then you can see that this is one simple classification, what maybe in the textbooks people give, like it's more of retrospective studies or prospective studies. And retrospective studies, again, it's more important. I'll not go into the details of the study design, but remember about there is an outbreak of any disease in your place. And if you go back and try to collect the data, we need to collect the data on disease and collect the related information using a questionnaire. And you can always do the descriptive epidemiology part. And this particular thing, we have organized many trainings in this aspect to and train people to collect basic data so that they can describe the disease in terms of the space and time. And the analytical component is also we can think of adopting when we have expertise in the field. And the outbreak investigation, which is the topic of today's webinar, is how we do systematic outbreak investigation, that I will cover in detail. And there are prospective studies also. This is one thing which you need to remember in a nutshell, or in a summary way of doing any epidemiological studies. Most of the times, what we observed or what we discussed with the field veterinarians or maybe the researchers is people think, this is my observation, like people think the outbreak investigations is very difficult task, or the epidemiology itself is a difficult task, because the way it has been, the subject has been taught at the UG level, it's very 1+1 credit, and and now the discipline has been shifted from one department to the other department, and that way the most important subject is being neglected, I would say very honestly, on this platform as well. So that there is a need for that. So, but the point I'm trying to make here is, if you do it with very basic understanding, you can contribute not only to contain the outbreak, but also prevent in the future, and so that we can not only control the outbreak and see that the farmers are getting benefit. Ultimately, the rural farmers which are dependent on their animals for their source of income and livelihood, they are protected. And ultimately, the other aspects now, because of all the COVID and other things, this is very critical component. And you talk of One Health also. And most of the times, people say that why we need to do outbreak investigations, right? This very first question comes to our mind when you want to study any or do any outbreak investigations. So very simple things, if you say that, like, we can do the outbreaks investigations, without any proper way, or you can do systematically, so that the main 3 points, if I want to highlight here, the source of infection, right? Where the animals got the infection. We most of the times fail to identify the source of the infection for that particular disease. So that is why, because we don't collect the data systematically, we don't try to analyze the data systematically. When I say systematically, it is not very high-end data analytical skills are required for that. Very simple, using simple, calculations, we can come to some sort, idea of the source of infection, so that it can be used to control the disease and also limit the disease spread. Most of the times we behave sometimes not doing anything. I'll take examples not only from the sheep and goat, but also from other species of animals, because there are many diseases like we could not contain in time. And the other important aspect is when we do systematic outbreak investigation is we can also evaluate our existing prevention strategies. Ideally speaking, when you have a vaccine for a disease, the outbreak should not happen. Like, there's some problem in our vaccination strategy, our containment strategy. When there is a single case of any disease, what we do? So those things will come to know only when you do some systematic outbreak investigations, right? And then finally, to prevent the future outbreaks. If you have these things in mind and do the investigations, you'll have enough evidence to say this is what can be done in the field. And what information it will give you, how the disease is spreading in the population, what is the spatial distribution of the disease, and what are the characteristics among the disease. I said about this point in the purpose of the epidemiology slide also. And what does that differentiate between, or what is that determining the occurrence of a disease among affected and non-affected group, and what is that risk factor? ultimately, if you identify that, so that what we can do is in terms of the intervention point of view, we can say, okay, this is the source of infection, or this is the time of the infection which animals are getting infected. I'll show you the examples when I try to cover in detail about that. So why do outbreaks occur? And this is again one question everybody thinks about that. But I would say that once you have to understand any disease, if you are investigating, this triad is important. Very fundamental triad of epidemiological principle, like agent, host, environment. When we investigate the outbreaks, we have to think about what is that agent, what is that infectivity of that, pathogenicity of that, virulence, immunogenicity, many factors of the agent. We need to consider that. And what are the host factors? What is the species of the animals? What is the age of the group affected? What is the sex of the animal, breed, nutritional status, physiological status? And these data, most of the times, we fail to collect. And the environmental factors. What is the housing management there? What is the stall fed or grazing? What is the weather in that place? And combination of these factors, or the interaction of these factors of agent, host, and environment will lead to occurrence of a disease. When these combinations of this interaction of these combination of three factors will determine the occurrence of a disease. So these things always you need to remember in a particular place when you see an outbreak, how this agent, host, and environment in that place is playing a role there. So how it occurs. So if you just try to see, what I'm trying to cover is why outbreak investigation is important. And in the previous slide, I saw why they say that importance of that and why outbreaks occur. And if I say how outbreaks occur, like what is the transmission pathways. I'm covering very basic fundamental concept which we all know and you all know also, like, but sometimes we fail to put it down and see that what may be the transmission pathways for that. This is standard terminology is given in different manuals also. What they say is why the outbreak occurs, or the how outbreaks occurs is the source of agent is required. Whether it can be from some symptomatic animals, so they get the infection and transmit to the other animals. It can be some asymptomatic animal, or in the animal which can be in the incubation phase, or a convalescent animal, or another animal species, environment. This source is important, right? So from somewhere the animals are getting infection. We need to be very, disease investigation is as good as doing some criminal investigation, as we always say that point, because we have to be with that kind of approach. We can identify that source of infection. And what is the portal of exit? When I mean portal of exit for this is if the organism is there in some animal as a carrier status, or the organism is there in the environment, from there it has to come out and cause the disease to susceptible animals. So that is more important. Once it has come out of that, then it has to transmit to the other animals. So that is the mode of transmission there, by which the agent passes to the susceptible host. What is the transmission mechanism for that? It can be direct, indirect, and for some agents, both pathways are important. I'll tell you an example of anthrax in the next slide, how the mode of transmission is important there. And the portal of entry. This portal of entry is with respect to the host barriers, whether it is by bite of vectors, or it is by ingestion, or by any other routes. So that pathway, how it enters the host. We need to understand these concepts. Then we need, when you are doing the investigations. So then we'll be able to identify, how the disease is transmitting from one animal to the other animal. So these are the very four fundamental points to remember when we talk of how outbreaks occur. I'll take an example of anthrax, how these 4 points fit into there. So everybody knows about the source of agent is most of the time, because these spores survive in the environment, and if there are some environmental condition changes, like there is flooding or drought conditions, which will sort of make the organisms accessible to animals. So that is your portal of exit. Once the organism has come on, maybe in the water form or somewhere it is there for the animals to be consumed, then the mode of transmission point comes here, like whether it's an ingestion mode or inhalation, insects, or other modes. So when you talk of anthrax, everybody knows about this. Ingestion is the main transmission route for the animals. Inhalation is more for human cases, but still that is also a possibility. Insect also play a minor role there. Other modes like movement of animals, infected animals to the other areas. I'll show you the examples how this transmission from one place to the other place can be an important factor. And the vultures and other wild birds is one important thing. And once you talk of the mode of transmission, then comes your portal of entry of the organism inside the animals. How the spores penetrate through the oral lesions, how the germination takes place, how the multiplication of organism takes place in the lymph nodes, and migration to other lymphoid organs. Splenomegaly is one important, pathogenic lesion for this anthrax, and septicemia and death. And this is what the outbreak, how outbreak occurs. If you want to take an example of anthrax, and that can be explained here. If you have many villages or one village, you see that outbreaks do not happen every month or every day. When does it happen, and why does it happen in particular point of the time? So there is something, these are general points to remember. It is not specific to any bacteria or virus or any parasite. There must be something, there is any exposure to the agent. That means some favorable conditions. You all know, like for many diseases, you have some temperature fluctuations or some rainfall fluctuations that will increase the exposure of that particular agent to the susceptible host. In some cases, you also find there may be some increase in the infectivity or virulence of the agent. Some new serotype has come, new strain has come. You all see now, COVID example is good thing there. So that is one thing to remember, like when it happens. Other important point is exposure of the agent to naive host or the animals which have never been exposed. So those animals are highly susceptible for any disease for that. And there may be some efficient way of transmission. Maybe it is some water source which is causing the outbreaks. We have to look into that. And whether there is increase in the host density, thereby leading to increase in the host exposure to that agent. And there may be some change in susceptibility of the host. One example is change in immunity status. That is one example for FMD people, say the vaccination duration of the immunity, and maybe for 4 months, and then the outbreaks will happen there. And maybe some new entries like movement of animals or any other factors. And these are simple points to remember. You have to always make it a note of your investigations. I'll cover in the systematic data collection part when we try to investigate the outbreaks. But these very simple things we need to consider when you are investigating, whether there is some increase in the exposure or the virus or the bacteria, there is more infectivity, or there is some susceptible population has been introduced into the a village, or maybe there's some immunity problem, or there's some vaccination that needs to be changed. These very simple points to be remembered. So with this, importance of the outbreak, how it happens, when it happens, and these factors determining these things, I'll try to cover the outbreak investigations in different steps. Before I go into that, the steps in outbreak investigation, I'll put across to you a very simple example in field what you observe day-to-day life, and then you'll realize the importance. What is the role of field veterinarians in that place? Ultimately, it comes down to field veterinarians and their role in doing the outbreak investigations, and also they becomes bridge between the policymakers and researchers. So their role is important. I'll put this one simple flowchart to say that there is an outbreak or a suspected anthrax case in. What field vets do? Maybe they visit the outbreak and find out the location of the outbreak, and they collect the sample, send to lab. Anthrax is confirmed by staining, culture, or PCR, or by authorized labs, and report goes to the higher level animal husbandry officials. One aspect which is lacking is we don't answer the question why anthrax happened in your place. We do this routine basis. This is what is happening. Samples are collected, confirmed, and then report goes to the higher ups. But we don't answer the question of why anthrax happened in your place. I covered all the points of importance of the outbreak investigations and how it happens, when it happens, and those things are lacking here. This is one thing we always remember, this point of, and where the field veterinarians play an important role there. With this background, I'll now maybe try to cover different steps in outbreak investigations when you're in field. And because one thing I have to tell you about when I talk of these steps in outbreak investigations, but remember this can be done with a limited training. So when I say steps in outbreak investigations, it's not like very difficult steps to follow. Put into different blocks. So when I say these first 4 steps, prepare to investigate, confirm whether there is an outbreak or there's some hoax call or something people have just said like that. Verify the diagnosis, find the cases, and perform simple descriptive epidemiology. Descriptive epidemiology is a simple thing like which animal got infected, where it happened, when it happened. This simple thing we can do with simple data collection. And the 6th and 7th point is developing hypotheses, evaluating hypotheses. Maybe sounds very too technical terms, but that can also be done. But up to 5th step, it gives a lot of information. And in particular, sheep and goat, where there are animals are there in huge number of flocks, we need to collect the information and count the animals affected and not affected. Even if you skip the 6th and 7th step, your role is important in implementing the prevention and control measures. And these steps are 9, 13. Number is not important, and not even the sequence also is not that important. You have to remember that we are doing outbreak investigation by covering all the points here. And how do you do that at the field level is important there. But while doing that, we need to remember like what the purpose of these first 4 steps are. Like when you talk, it's mostly related to counting the cases with counting the animals with the disease and without disease. That should be your motto when you do the 4 steps. Whether you do all these things together in all the 4 steps at a time, you can do it. But remember to collect the information on the outbreak with respect to animals which are affected, and also healthy animals data is very important. The comparison part is very important here. That's what they always say, that compare, count, divide and compare. This is a CDC concept. They say like always when you're doing the study, the CDC, you remember that count and then divide and then compare. Comparison between healthy animals and affected animals, so that you can know why. Any outbreak investigation, people who are in field, that if there are 100 maybe 50 flocks, or maybe you say 50 households with animals, not all 50 households will get the disease. That point you have to remember when you are doing the investigation. We have to see the information from the healthy animals so that we can compare why it is happening in the certain group of households or animals. And for the performing descriptive epidemiology, of your data to be described in terms of time, place, and animal, when it happened, where it happened, and who got affected, which species of animals got affected, what is the age group of that. So those things are important. Like in simple terms, if you remember, it is who, where, and when. That's, if you can remember, very easy to remember also, right? And the hypothesis point is to do more of analytical epidemiology there, why it happened. And most of the times, I would say that for the field level, you may not try to answer that also. But most of the times, if you perform the descriptive epidemiological steps, you can come to a conclusion about that outbreak, and even prevent or maybe control the outbreak, and also have your own prevention strategy for containing the outbreaks in future also. And the why part, we need people from the even that can also be done by, if you have a maybe 3 months training program, or do more systematic studies. And ultimately, prevention and control. Communicate your findings. Whom you're communicating findings. I'll cover that part in the last few slides. How to communicate your findings to, if you're communicating to media people, if you're communicating to farmers, if you're communicating to higher ups, how you should be doing that based on this data. Most of the times, we give very, when I mean we, like whole of our veterinary community, we try to not give statements based on some evidence. That's what the point of all this outbreak investigation point. To start the first 3 steps, how to prepare the investigation. You all do that. I'm just bringing in this kind of steps, how to do that. When you say preparation, always you need to have a checklist. So I think the audience who have been working in the field, or who researchers, for everyone, I think we always need to have a checklist of the things required for that outbreak investigation. All the sample collection kits, everything you need to have a checklist. And if it is some disease, zoonotic disease, or diseases like FMD or avian influenza, this is a practice for avian influenza for sure, but not for the other diseases. What is the PPE required? What is the minimum PPE required? And GPS unit, and I think we covered this part in the training also, how important is to have a GPS. It's very, very important. I'll show you the examples, how we collected the data and made maps of that. And the charts, other things are required when you want to make some participative village maps. And questionnaire is one thing, it can be detailed, it can be prepared, or basic data collection format, which covers the information related to animals, and which covers the information related to time of the cases, and which covers the information related to the space or the village. So the time, place, and animal components. If you cover the data in one-page format, it will be very good information to summarize your data. So this is when you're preparing your outbreak investigations. You make sure you're doing in a very systematic way. You need to have a checklist, and the team is important here. So who should be visiting that? We need to have a composition, whether if it is some we can have veterinary pathologist, microbiologist, parasitologist, or and maybe even veterinary extensions people also important when you're trying to collect data. At the field level, you can have your own team also who can do what work in field. If you have expertise with the different specialization, you can take them. If you don't have, still it is fine, because all these things can be done by people who are at the BVSC level also, if you want to properly follow these things. Other more detailed studies can be done by more experts with masters and PhD or researchers, scientists, whosoever it may be. Remember, these things can be done by field level veterinarians. When I say confirm, it's not something related to diagnosis point of view, to just make sure. Maybe some person will call you, there are a lot of animals dying in that. We need to ensure or ascertain whether there is really something of an outbreak happening in that place, or there's something, just people have a hoax call for that. The definition of outbreak is important here. And is increased incidence of a particular disease condition. And increased, when you say, it is always, to have a compare with baseline. Always collect information on the numerator part, number of cases, and the denominator is your number of susceptible population in that place. So if you forget to collect the denominator, you will not have. For example, I'll tell you, like if you visit a village where there are 100 animals, and if you find some 10 cases, so 10 cases, if you tell, then people will say, okay, 10 is more or less. It's very difficult to say unless you have your denominator there. 10 by 100 into 10%, then you can say that this percentage of animals are getting infected. So if you just say numerator, we say 100 animals, but 100 animals with respect to 10,000 is very less number. But 100 animals with respect to 200, then it is a very high number. So those things are important when you're collecting the data. So susceptible population data is important. And then why this is important when you're trying to say an outbreak is happening, maybe one case, we cannot call it as an outbreak. It may be some sporadic case of some disease. So that outbreak always will have more than one case in that particular place. And for the verification part, like when I say verification of diagnosis, the points I'm covering here are very basic points, which we don't need for at least for the clinical science and epidemiological findings. You don't need any lab support also. I'll tell you how it can be done, how we can use the clinical science and history, how we can use the epidemiological findings, and finally the laboratory-based confirmation. So these points are very important to remember. I'll take examples for each thing, how we can do it when you're in field to verify your diagnosis. For example, if you take clinical diagnosis as one criteria, then sudden death for anthrax. I will take the example for anthrax. One disease, if you want to take an example, I'll just use anthrax for all this discussion for today. Sudden death, everybody knows, and other symptoms are important. Like after death of an animal, everybody knows the unclotted blood exude from body openings. Animal body may not stiffen. Delayed rigor mortis. These are very simple points if you remember in the field. Other signs are important. And recent history collection is also important. And maybe in case of anthrax, we have noticed most of the times there is some disturbance in the land. There is digging, plowing, or something which has unearthed the spores, and then maybe some flooding, and animals get infected. I'll just give you examples of clinical forms, because these things are important when you are doing the investigation. For example, in cattle and sheep, what are the forms? When you say per acute form, we have symptoms like staggering, dyspnea, trembling, collapse with convulsions. Acute form, again, fever, temperature, bloody discharges, and localized subcutaneous edema, swelling, ventral, neck, thorax, and shoulders. Why I'm showing this particular slide is, when you're visiting outbreaks, these things are important to consider. These are all, whether it's a disease is per acute or acute form. And if you compare the other species, you have to see that what other species are affected, like, whether horses will have the disease, and pigs, dogs, cat, and wild carnivores, these are relatively resistant species. And this is for anthrax, but for all the diseases, we need to have that susceptible species which can be affected with that disease. And wild herbivores, the disease is similar to cattle. And the lesions, we all know, like these are the lesions of hemorrhages of various sizes, edematous blood tinge effusions. And these all things are to be noted down when you're doing the investigation. So you all know that. And when you're doing the investigation, one point is very important to consider is your differential diagnosis. I'm taking an example of anthrax and what we should be differentiating from different other diseases, for example. If you say hemorrhagic septicemia, I'll take the example when I cover that point. And most of the times it's sudden death and widespread hemorrhages. So that can also be observed in sometimes hemorrhagic septicemia also. But remember the points which differentiates HS and anthrax is splenomegaly. Similarly, like black quarter, the rigor mortis point is there, which is absent or delayed in case of anthrax, which is not there in acute black quarter. And it will be some non-infectious cause, so we have to rule out that. Acidosis, bloat is one important thing. History of feed consumption we can take. lightning strike, like longitudinal fractures, they say. So these are very differential diagnosis criteria to be followed. Like similarly for baby shows, tick infestation is important. Phosphorus deficiency, also you find hematuria and hemoglobinuria. We also find sometimes in anthrax cases. So we need to understand the tucked up abdominal appearance is the differential diagnosis criteria. Plant poisoning can also be there. Why I'm covering these points are like, when you're doing the investigations, we need to cover the diagnosis at the field level, what can be done. And you have to differential diagnosis and come up to say that clinically, it may be a case of any X disease in field. So I'll take example, when we did the investigations, how we try to arrive before taking the samples and bringing it to lab and testing it. So this is what we do it in field, for example. This is one example in sudden death in cattle. So more than 22 cattle died in that village. And in most of the cases, it was sudden death. So what were their observations like? Sudden death, we did notice the absence of rigor mortis, there was bloat, and there was a history of anthrax in neighboring village. So this is one thing we can narrow down it to anthrax. This is a very important study which we investigated was in sheep. There was bleeding from natural orifices. We see that photograph also, and hematuria was also there. And 50 sheep died in 2 flocks. Sudden death, bleeding from natural orifices, and differential diagnosis we consider was plant poisoning. And this is one more interesting example when we try to collect the history of that. Based on the history part, host species was sheep, and more than 15 sheep died from a single household. Why I'm showing this is, when you try to collect histories, I'll show you in the photographs also. But the village has history of anthrax. So the history taking is very important here. So when we investigated, we found that the person who's standing there, this is the place where the animals are, the carcass, the dead animal carcass are buried in this place. And there is history of anthrax in this village. This man, what he did is he took this soil from this place where the animals were buried, and maybe after 2-3 years, there is no sign of any animal carcass there. But he took that soil and he brought to his place where he was constructing his house there. And then we found that only in that particular household, the sheep were dying. No other households were affected. And then when further we confirmed collecting the samples, and then vaccination was carried out to contain the outbreak. So this is what we need to collect the history, right? And the observation was recent disturbance of the soil where carcass are buried. Soil was also used. And for the differential diagnosis, this is one interesting study we got it in case of HS. It was diagnosed as HS. Regular, if you see the pattern of death there, every 1 or 2 days, deaths were happening. And it was confirmed as HS, and HS vaccination was also carried out. But again, the mortality continued. Buffalo died, cattle died, one more cattle died. And then we visited and investigated and took the history. And one important sign we observed was there was delayed rigor mortis, if you see there, and bloating of the carcass. And with that, if we closely observe, there was also bleeding from the natural orifices also. These kind of observations, if we do and supported with the lab, we can confirm the outbreak, verify your diagnosis with that. This is one more case of outbreak, there also some history of soil was brought from the other affected village. So if we can observe this map, we find the cluster of cases very concentrated in this particular region, and somewhere the soil was brought in this place, and cases were happening in this particular cluster of households. And in this outbreak, sheep, goat, cattle, buffalo, all the species were affected. So when you're doing the verifying your diagnosis at the field level using clinical symptoms, this is one more study we did, or when we investigated, we found these were challenges we faced in field, and taking those examples. When you took the differential diagnosis, have to consider enterotoxemia or other clostridial organisms. There were again 20 animals died, 4 sheep and 16 goats. Mostly the deaths were sudden deaths in goats. And blood was not clotted in few cases. Anthrax, we all know it's very rare in goats. So, but what we got is this particular case was, there was some staining. We know, everybody knows the anthracoids, which we get in staining the gram-positive organisms. Based on that, and it was sort of declared as anthrax, but it was not case of anthrax. So these are the things we need to consider when you're doing the verifying your diagnosis based on clinical science, epidemiological findings, or laboratory-based findings. So what I've covered is, just basically when you're doing this routine investigations of the outbreaks, I'm just trying to bring in how you need to confirm it and verify the diagnosis based on field observations, the differential diagnosis based on history, clinical science, and I'll give you the examples. Once you know a village has been affected with any disease, we confirm that, okay, this is what is happening in that village. we have verified the diagnosis and verified the diagnosis based on the clinical symptoms or based on the laboratory findings. And we know this village is affected with that disease. The most critical part, and which is not been followed in field, is finding of the case. The initial few slides itself, I mentioned about we need to collect the data from the animals which are affected and also the animals which are not affected. Case finding and data collection. When I say case finding, is the most critical step in your outbreak investigations. How you find the case. For example, if you're visiting a village and you have done all the things, we know there's an outbreak of anthrax in sheep, for example. But how do you collect the data? How will you find the cases? Just randomly collect the data from the any household? How you say that this particular case or household or flock is affected with anthrax? How you define that case? For that particular purpose, the point of data collection, remember to collect the data or find the cases using proper case definition. If you don't do that, most of the times you will not be able to classify the cases and controls. And the village, if you're visiting that place, we need to classify each and every case as affected with the disease or not affected with the disease. And in order to do that, we need to follow case definitions. And that's what we are trying to push also to maybe the authorities also. And we are covering this aspect in the training also. How you define a case. And most of the times what happens is, if a village is affected with any disease, HS, anthrax, BQ, FMD, or any disease in sheep and goat or any large animals also, one sample is collected and it has been confirmed as any particular disease. But what about the other animals? How you classify the cases? For that, to collect the data from the healthy and affected animals, you need to define the cases. So if you take the case definition part, we say the clinical case definition. And the veterinarians are the right people who can classify the case to say that it is the suspected case of anthrax. And we know the symptoms for anthrax. If there is sudden death, take the history from the owners and try to classify the case. And if you do the postmortem, I have written it in bold, like should not be open. The carcass should not be open in case of suspected anthrax. If accidentally open, then you can observe the dark unclotted blood, which is characteristic to anthrax. Finally, the laboratory test of gram-positive rod, Bacillus anthracis from infected blood or tissues, growth of the bacterial blood. With this criteria, which is stage 1, stage 2, and stage 3, if you visit a village and if you collect the information, you can classify the case and collect in one-page format and say that this is a suspected case of anthrax, if it satisfies the clinical case definition. That can be done at the field level. I think everybody agrees with that point. We need not even depend, because once the outbreak has been confirmed and verified based on the laboratory findings, you can visit and collect from all the households which are having animals and try to classify the cases. Whether this household, you can classify based on the household or based on the animals also, and classify based on the clinical case definition. And if probable case also is there, but if you just remember suspected or confirmed, these two criteria are followed. For example, if there is some cases, it may be the case of FMD or some important disease, each and every animal samples are collected and tested, then you can use the confirmed anthrax case definition or any other disease. If you don't do the confirmation for each and every animal, you can always use the suspected. And why it is important is when you're classifying the cases, we miss most of the information because we don't know about the other animals. So classify the case based on the clinical symptoms and suspected case of anthrax or any other disease, and then collect the data. So why it will be helpful, I will tell you in the next few slides also. When you try to describe the data in terms of time, place, and animal, this will be helpful. So once you have done the first 4 things, basically, if you say 4 steps, basically we didn't do anything. One thing is we have verified the diagnosis, clinical symptoms, differential diagnosis, everything is done. You're finding the case. For example, there are some 100 animals, you have collected the data from each and every animal from that village and classified that whether there is a clinical case of that disease or it's healthy animal. Once you have done, then you can summarize your data and say that time, place, and animal component can be put. I'll briefly explain you how this data collection, and that's the most important component of the outbreak investigations I'm covering now, how these things are important in terms of time, place, and animal, and how this is useful for prevention and control ultimately, right? So for the time component, we call it as epicurve. I've heard of it, but it's a very simple thing. If you have some pen, paper, or maybe Excel, you can do that part. It's just a visual display of when the disease started in an outbreak. So the shape of the curve will be determined maybe based on the incubation period of the disease. So you should be completely, once your diagnosis is confirmed, we should be very clear about the incubation period of the disease, what is infectivity of the agent, and the proportion of the susceptible animals. Because some diseases, the infectious agent is a highly infectious agent with short incubation period, with high density of susceptible animals, we can have a sharp rise in the cases. So how it can be done, how the epicurve can be plotted, we'll just take you very simple examples of that also. So basically an epicurve will have your x-axis, your y-axis. In your x-axis, you have date of onset of the illness. There are some guidelines for this one, the CDC website also, you can see that. And the x-axis is the rate of illness and y-axis number of cases. If you plot that, you can give an epicurve. Mostly make sure when you're doing this kind of things, when you're equal intervals, 1, 2, 3, 4, like how these intervals can also be calculated, I'll show you that. So this simple plot of your data. So when it can be done is when you visit the field with simple format, day 1, first case was reported, and day 10 or 20, the whole outbreak period, for example, last case of the outbreak, first case of the outbreak. Collect the data for 20 days, for example. This particular outbreak, for example, it lasted 20 days, and plot in this one. So each day, how many cases were there? So we can just get an epicurve of that. Once you plot that epicurve, what information it gives you is, if you see the first graph, is your point source epidemic. So if you can see that it suddenly rises on one peak, and this particular epicurve will show you that there is one common source of infection which led to this point source epidemic. And food poisoning is a very classical example of that, and anthrax also. If you know about anthrax, some contaminated soil or water, it will be source of infection, and all of a sudden many cases will be there. And if you plot in this manner, what I just explained you, and based on the collection of the data from day-wise collection of the data, then you can know whether it's a point source epidemic or a continuous common source epidemic or propagated. And when you say continuous common source, is like the source of infection is always there. It has not been removed. That's the reason it's continuously happening there. And propagated outbreak is something which is foot and mouth. This is a very good example of that, because once you have that, the disease is transmitted from one animal to the other animal. So that way we can, when you plot the graph, you'll know what kind of, so this will help you to come to a conclusion whether this is an anthrax, a FMD, or any other disease. And I'll just maybe tell you how this picture will help in maybe just one slide. Is what we get is most likely period of exposure of that particular agent. So we know peak of the outbreak, we know minimum incubation period, we know maximum incubation period of that average incubation period. Forget about everything. If you plot that, and if you see that area and the gray shaded area, once you plot, we can come to a conclusion that, okay, this particular period was the likely source of infection for that particular outbreak. If you remember this point from the epicurve, that is good enough. So when you plot, then only you'll come to know. For example, as I said, if you have 20 days of the cases reported in that village day-wise, you plot it and calculate based on the incubation period, we can know when the animals might have got infection. So then we can go back to time, what happened in that period, where there was some introduction of the animals, what many other things you can consider in that period. So this will be very helpful for your identifying the likely period of your exposure. This interesting example I'll tell you, which we did, was when you're collecting the data from the day-wise. And this is continuously, the outbreak was happening one place in Bellary actually. As the 24th August was the first case of anthrax. 25th August vaccination was carried out against anthrax again. But if you see that again, after so many months of vaccination also, 26th October to 29th October, still there were cases of anthrax happening in those sheep flocks. So when we visited, again, anthrax was confirmed in 27th November. If you see the time period here, August, September, October, November. So 4 months the outbreak was happening. So we collected the data from that particular place and tried to identify what is happening in those months, what is happening in August, from 25th August to 26th October. So when we collected that data, so in that period, the antibiotics was used. So in a way, when the vaccination was given, along with that, antibiotics were also used, leading to failure of the vaccination there. And that is the reason the outbreaks continuously happening. So this is what, when you collect the time data, we can help you in identifying them, some what can be intervention can be done. And then we advised, there are some guidelines also. We should not be vaccinating using any antibiotics for 10 days after vaccination. And those things should be, because when the animals are dying, farmers use antibiotics themselves. And this is what's importance of collecting the time data. And all this data can be collected in one page. I'll repeat this point. There's nothing which you're collecting some additional information. And when you're trying to describe the disease in terms of the animal components, like when you record the age of the animal, what is the breed of the animal, what is the sex of the animal. This is very important because there are certain diseases which are age specific, some breed specific risk factors are also there. Which are sex specific risk factors are also there. And this information will also be helpful in categorizing the cases as exposed and non-exposed. And then we can identify the source of infection and risk factors for the occurrence of the outbreak. So the animal details we need to collect whenever you're visiting the outbreaks. What we can do with that data is, we can calculate, this is a simple formula for attack rate. We can calculate mortality rate, morbidity rate. It's not very difficult to calculate also. When you have data collected for some 20 days, for example, day-wise, then just plot it in there. You can do it in your calculator also. And then species-specific attack rate can be calculated, age-specific attack rate can be calculated, breed-specific attack rate can be calculated, and case fatality rate. Just remember how it can be helpful in outbreak investigations if you collect that data. So there is some related information, there is some information on the age, and there are other information which is related to the animal details. I'll take a few examples of what we did. For example, there were 15 deaths, and the total animals in that flock were 30. Then we can calculate the attack rate of the village and the flock. Moving on, when you're collecting the information, place is important role. And we train people of how to use GIS and GPS in mapping of the outbreaks. And as I said, this is just an overview of outbreak investigations. You need to remember, but still all these things can be done. And maybe the GIS component cannot be done without any training, but still remember that can also be done. But if you do, even hand-drawn maps can be done when you have some difficulties in doing that. So we train people for 5 days training program that will be covered. So, but I'll take examples how it will help in your outbreak investigations when you map your outbreaks. This is an example of Bellary district. This is one thing which we investigated. There are some flocks, sheep flocks here, and this is a grazing area, and this is a source of water body for that animals. And this is complete area which is far from the village. All the flocks are kept in this place. When you collect the data, we can map it out and see where it is happening and what is the distance between the flocks, and many things we can understand from that. And this is another example of when you collect the space data. What happens when you're visiting outbreaks, and if you visit without any latitude, longitude information, we fail to appreciate this point. If you say these 2 flocks, which are around 2 to 3 kilometer distance, and these outbreaks were happening in both the places, and both got confirmed. So when you map it out in this manner, then we can see how the outbreaks are spreading from one place to the other place. And just giving examples of what we have all done also. And this is one more example of in Chamarajnagar. So one village reported first time anthrax, and the neighboring village reported regularly. In this map, if you see that, first village is endemic to anthrax, which is Sindhanapur. And this village, because it is endemic, they carry out the vaccination regularly. When we visited, this is the village which reported first time outbreak. So this is one thing interesting when you try to map it out on what is the distance between these 2 villages. It's not hardly 1 or 2 kilometers. Then definitely there is a movement of animals and people happening between these 2 villages. One point I have to highlight here is we need to have a vaccination strategy to say that we need to vaccinate in other places also, not only the affected village. So when you use the place data and try to map it out, we'll have a lot of information, try to identify the source of infection, and we can also know that what is the spread of these diseases in that place. And this again, I showed you the clustering of the cases can also be identified when you use mapping and collect the latitude, longitude information. This is very simple exercises. We can do it also. And this is one outbreak investigation we did in Harihar Taluk of Davangere district. If you notice in this particular map, some red dots are there in all the places. Now, this particular place was a place where the outbreaks of anthrax was happening. The place which is just in yellow circle here. So when we visited that place, we could not identify the source of infection. Then we found that there is one place in this area where the migratory flocks come. And this village, the sheep population from this village also mingle with this migratory flock. And that's the reason they got the infection from that. And then again, we went to other villages nearby. Wherever this migratory flock were visiting, we visited those places and we could find the cases were happening in all the places. So this is what happens when you try to collect the data in space and using some simple tools like GPS and map it out. And this can be done in 5-10 minutes also. But just to remember, these are very useful tools, especially in sheep and goat, because the numbers are more. Migratory population is also there. We know it can always be helpful there. This is one interesting map. When we interacted with the migratory farmers and we tried to collect the data, we mapped out and asked the farmers to name the villages which they have visited. We see their migratory pattern also started from one place and went to other village, then next other village. They're moving in these places, and then finally went back to their place. And we found the outbreaks were happening in there along their migratory track also. So this is what the history we collected. This we mapped it out and we collected the data, and we also confirmed in the lab also. And this is the importance of the space when you try to collect the data. I'll repeat the points what we have covered. We confirmed the outbreak, verified diagnosis, collected the case data. Based on the case data of affected and not affected, we performed descriptive epidemiology. Which animal got affected, where it happened, and when it happened. And this can be done with one-page format. I'm saying, repeating that point of one-page format. Okay. And the point of 6th and 7th, which I'm covering here, is which can be done, which we have done. I will show you in one example of that and how we did that interesting study also. And to know, to make people aware, like this can also be done when we have some good data we collected. So the problem in that particular case was, I'm talking example of how we try to identify the risk factor point of view. And this particular point I'm repeating, you may do it or may not do it also in field. Maybe we can take help of people in our institute or maybe your colleges nearby. So the problem in that particular village, there were some 6 villages, there were some outbreaks of anthrax was happening. And so our question was why it is happening in few flocks of the villages. So we developed the hypothesis and tested the hypothesis by using a questionnaire. Questionnaire is nothing but we collect the information in systematic manner based on our hypothesis, what risk factors may be important there, and analysis of the data, identification of the risk factors. So this is what the point. So to answer the why question, we did this study. And so that we can prevent, not only we can have a prevention strategy also. So what we did was we collected the data from 6 villages and collected from different households and try to identify that what are the practices they are doing in that particular households. Household, I mean, be like different flocks in that household. What kind of practices feed? We maybe collected the data from using some 20 odd questions there. So we found this is the descriptive statistics of that data. Villages affected were 6, and the total flocks in that villages were 102 flocks, and total number of animals in 6 flocks were 9986 animals. Households affected, that is 16% household, and the mortality rate of 2.5%. This is what we can collect the data. But interesting point, what we have noticed in that particular investigation was why only few flocks are affected was size of the flock was important. Like the size of the flock is more than 100 animals. The risk of getting the disease is 4 times compared to the flocks of less than 100 animals. I'll repeat this point because this is one thing when we talk of risk factors, we always estimate in that way. So that we identified it. And this completely the evidence which is done with some data analysis. So flock size, more the number of animals, because ultimately spread was more. And animals were moving from one place to the other. So the risk of getting diseases in flock size of more than 100 animals is a 4-time risk. When you say this point is very important, right? So the risk of this getting disease is 4 times more. And the practice of migration. So wherever there is flocks which are migrating, there the risk of disease is mostly like around 3 times more. The animals which are stall fed or which are on the local village level grazing is happening there. So why? Because when the animals are moving from one place to the other place, not only they are getting the infection from other villages, but also spreading to the other village. And this is slightly something we need to check also. We can have some migration is also important. It cannot be completely discontinued. We need to check these things. And last important factor which we found was very interesting was people are vaccinating after the outbreak. So that is also the more risk of anthrax was because they were doing as a reactive measure, not as a proactive. So the risk of such thing is, we found that 4 times the risk is more in that kind of flocks. And compared to the flocks which are vaccinating before the occurrence of the outbreak. So this is again interesting finding. So out of some 20 odd questions, we found these points are important in the complete study. So again, awareness, prevention of migration, checking the migration, vaccination status, many other things can be reduce the burden of this disease. So whatever we do, ultimately we need to adopt a strategy to prevent and control, right? So we cover this in our trainings also, how to make a zone of 5 kilometers, 10 and 15 kilometers buffer zone. And these are very important tools also when you try to contain the outbreak. So based on the disease, and again, the criteria to define 5 kilometer, 10 kilometer, 15 is also vague. For each disease, we need to define that zone. So then we contain very systematically in those zones, we can prevent the movement of animals, we can carry out the vaccination, or we can carry out the surveillance. And all these factors can be, considered when you're making zones in that particular place. So finally, when you're trying to, you have done your investigation, either descriptive study or your analytical study, implemented the control measures. So we need to prepare an outbreak investigation report and also communicate your findings to people. So your audience can be, when you're preparing an outbreak investigation report, you have to summarize your report in descriptive epidemiology point of view, which animals got infected, what was the mortality rate, case fatality rate, and when it happened, what was the likely source of exposure on a time, what was the source of infection, where it happened, whether there was a clustering of the cases. All these things can be put in your report. And you are communicating your findings either in terms of report or by using some briefings to the people. So you need to communicate to farmers. The farmers, what we communicate is not about the risk factors or about any other descriptive epidemiology, right? You need to communicate to farmers to say that, how best you can prevent the disease by vaccinating on time. Migration can also be done after vaccination, or maybe checking the migration also. And we need to explain about these, size of the flock, how many other managemental practices can be explained to farmers. And when you're talking of veterinarians, your fellow veterinarian colleagues or any other people, we can communicate in a different language. We can present your all scientific findings. And for the policymakers, we can advise some policy level decisions. All these reports or the communication should be different. I'm not going to the detail of this communication part here. It's completely different exercise altogether. So the report should also have simple format, summary, introduction, methods, discussion, what lessons we have learned from that, what is the recommendations, and other things should be covering. These simple things you need to prepare a report then again. So finally, to summarize what I have discussed in this webinar is, although it looks like different steps, but just remember what we have to do when you're in field is just to verify your diagnosis, whatever the knowledge you have. And the most important point is how you collect the data and based on the case definitions and finding the cases in a village and summarizing the data in terms of the animal, place, and time. As I said, if you collect the data using one-page format, we can make summaries of that particular outbreak. And the hypothesis part I'm not covering here, it's fine, but remember that is also important when you say risk factors. What is the risk of getting infection or outbreak? And the prevention and control. Unless we have a prevention and control strategy, all the outbreak investigations are of not use, not much help to the field or the farmers or policymakers anyway. But remember the importance of this particular topic. And so that at least if you have any doubts, any concern, you can contact us anytime for that matter. Any outbreaks investigation, we'll be happy to provide our services. And remember to follow this, at least simple case definitions, how to do differential. You all do that, but do it in some proper formats to be collected, and so that you can address very basic questions in epidemiology. Thank you. Thank you one and all for your time.